The NGS module in Qlucore Omics Explorer enables exploration and analysis of sequencing data, such as DNA-seq, RNA-seq, and ChIP-seq data. It runs locally on your PC or Mac with no need to upload large files to the cloud or a server. Researchers use this module for both qualitative and quantitative analysis. It includes an interactive Genome Browser supporting advanced genomic analysis, including mutation analysis such as variant calling, and also detection and analysis of gene fusions for RNA-seq data. For variant analysis, you can choose to either run a variant calling algorithm while loading your data or upload your own VCF file to filter on variants. The NGS support also includes peak detection for ChIP-seq and ATAC-seq data analysis, allowing the user to quickly find regions of interest such as promoters, structural hotspots, and pseudogenes. Additionally, matrices for RNA sequencing counts, as well as detected peaks, can be generated using a GTF file for a comprehensive analysis, and you can quickly swap between the genomic space and expression-based analysis and use the broad suite of easy-to-use statistics available in Qlucore. Qlucore's NGS module allows the user to interactively visualize genomic data and supports research in comparative genomics, epigenetics, and transcriptional regulation. To learn more, visit our website.